Thank you very much, Christian, for the introduction. And the title of my talk is Modeling of nano-amperometric measurement of sub-quantal glutamate release by living neurons. And as follows from the title of the talk, here we're interested in the release of the glutamate, okay? And the glutamate is the most abundant neurotransmitter in mammal nervous system. And another important property of glutamate, which is apparent for this symposium, is that the glutamate is involved in excitotoxicity. And to set up in context, we consider in the process of the communication between the neurons. Okay, and when the neuron is fired, the nanometric vesicles, they are forced to fuse with the plasma membrane with the creation of the fusion pore through which the neurotransmitter can diffuse into the synaptic cleft and reach the postsynaptic receptors in order to propagate the signal. And to finalize the neurotransmission event, the glutamate, which is in the synaptic cleft, should undergo uptake, okay, that by the neuron through the special glutamate transporters, or by the glial cells, which then refuel the glutamate in the form of the lysine back into the neuron. And up to now, everything is fine. However, if something going wrong, for example, the transporters are not sufficiently, I would say, efficient, or otherwise, if the neuron is excited too much, we have a kind of accumulation of the glutamate in the synaptic cleft, and this has the toxic effects, okay? And these toxic effects are obtained through two ways. So first, it increases the oxidative stress inside the cell, and this is because the glutamate affects with the NMDA receptor, and which one is mostly permeable to the calcium ions. So this basically leads to the increase of the intracellular calcium. And this already activates several other pathways, including the nitric oxide synthase with the creation of the nitric oxide and the peroxynitride, as it was explained before, as well as affecting the correct work of the mitochondria, which increase the production of the reactive oxygen species, which induce the damage to various components of the cell and may lead to the cell death. And in addition, the very high concentration of calcium can activate the calpain's pathway, which can also lead to the programmed cell death. And in addition to that, the increased glutamate concentration, they lead to the weakening of the cell defense because the superoxide dismutase is inactivated, and also that the antiporter, the glutamate cysteine, reverses direction, and this sends cysteine outside of the cell, which eventually lead to the decrease of the glutathione concentration and with the evident effect on the ability of the cell to scavenge the free radicals. And as you can understand, the consortium which I represent here, we are great adepts of the electrochemistry, and so evidently we would like to study this kind of effects, but also the vesicular exocytosis, which at the origin of all this story. And this is because the electrochemistry propose several advantages, okay, for the monitoring this kind of events. The only problem in this particular case is that the glutamate is not electrochemically active molecule, okay, and for that, our collaborators fabricate the enzymatic nanoelectrode, which is similar to what was just presented by Professor Amator. This is the one which consists of the silicium carbide, and on which the successive layers of the carbon and platinum is deposited. And on the top we have the layer with the glutamate oxidase. And finally, this electrode also have relatively small sizes, which allow to perform measurements outside of the cell, but also to go easily inside of the cell. And the basic principle of this sensor is the following, that the glutamate is oxidized by the glutamate oxidase by producing the alpha-ketoglutarate and ammonia and creation of the hydrogen peroxide, which in turn can be oxidized on the platinum surface and lead to the detection current. And already with this tool, okay, we can characterize exocytotic events. In particular, we thinking to characterize the exocytotic pathway in dynamics, but here the important problem is the problem of the quantal or the sub-quantal release of the glutamate. And this is a very old problem because it stems from the early ages of the neuroscience, where it was believed that every exocytotic event should end up in the full fusion. So when the membrane of the vesicle fully introduces into the cellular membrane, okay, and this leads to the release of the all vesicle content. So basically this, from where it goes at the quantal size, that the vesicle is a single quanta. Okay, but there is a mounting evidence that the partial fusion is an important exocytotic pathway, and in this case, the fusion pore remains relatively small with respect to the vesicle size, and also that in this case, only some fraction of the vesicle of the neurotransmitter content which is released to the outside, okay? And also this kind of thing can affect the toxic effect of the glutamate, okay, if we can understand these things better. And to characterize the quantal or subquantal nature of the glutamate release, okay, the two types of experiments were done. One is at the, on the top of the varicosities. This means that the electrodes stay outside of the cell, and in this case, one can estimate the number of the molecules released during the vesicular exocytosis event. And on the contrary, with the intracellular measurement, where the vesicles collapse on the electrode surface, one can estimate the total content of the vesicle, okay? And here is the experimental example, the micrograph, okay, of the electrode over the top of the varicosity. And so this is a typical trace, and all the, I would say, zoo of the spikes observed in the experiment. And the important point here is that when we integrate the spikes to obtain the charge, basically the loading of the vesicle, we see that on average, the amount of the molecules released during the vesicular exocytosis is significantly smaller than during the intracellular. This is already a strong argument for the subquantal release of the glutamate, okay, enforcing that only part of the vesicle is used for a given event, okay, in exocytosis. And to characterize this case a little bit more, okay, we want to understand how much we are quantitative, okay, during both type of the measurements, okay, and for this we resort into theoretical approach, and the mechanistic equations, okay, describing the work of the enzymatic sensors are given here, okay, so again this is the conversion of the glutamate into the intermediate product with the creation of the intermediate form of the enzyme. And by reacting with the deoxygen, the intermediate form of the enzyme converts to the initial form with the creation of the hydrogen peroxide, which can be detected. Okay, and this one can be conveniently put into the kinetic scheme, okay, used in the enzymatic analysis. And we write it here in such a form which is used in the literature. Okay, so we didn't invent, and so we wanted to use this mechanism for the analysis. And since very often this kind of situation is oversimplified in the literature, okay, and discussed in terms of the Michaelis-Menten mechanism, okay, where the substrate is considered directly converting to the hydrogen peroxide, okay, emitting all the intermediate steps. Okay, so we will also consider it briefly in the following. And as you noticed here, so this mechanism is quite complicated and contains a lot of kinetic constants, okay, so information. So basically we need to extract all these information from somewhere to populate our model in order to be able to simulate this kind of situations. And our approach was relatively simple. Okay, so what we wanted is to we get the calibration curve through the experiment which are performed at different glutamate bulk concentration. We would like to model this calibration curve, extract the kinetic information, and then with this kinetic information we wanted to model the release events and try to reproduce the spikes observed in the experiment. However, this was not simple. Okay, I'm going a little bit forward, and there were two big problems, okay, which we had to solve, and one of them is related to the calibration curve, and in particular to the case that very often in the literature the two types of analysis are confused. Okay, and so when enzymatic analysis is performed, is generally performed in the enzyme which is just in the bulk solution, okay, and what people measure is the initial rate. So this is the variation of the product, or the same is the substrate concentration with time, and what they take is only the initial rate of the reaction at the very, very small times, okay, and this is done to ensure that the individual diffusion layers around each protein are very small, so they are not overlapping. And in this case, it's ensured that the reaction is controlled by the bulk concentration. And that's why all the parameters which we have here, they are depending on the bulk concentration of the substrate. And the two figures of merits which generally extract from this kind of plots is the, okay, I call them P1 and P2 here, which is the slope at the very small concentration and the asymptotic value at the very large concentrations. And for these two mechanisms, they can be written, okay, from the enzymatic textbooks, okay, they look like this. And if we can extract these parameters from this curve, we can then easily relate it to the enzymatic rate which we basically need. And in opposite to this situation, in the electrochemical measurements, very often people do this kind of analysis straightforwardly on the calibration curve obtained with the enzymatic electrode. However, this is basically wrong, because in electrochemical measurements, generally the measured value is at the quasi-steady state, okay, at sufficiently long time to ensure the full sufficient development of the diffusion layer, okay? And in this case, if we look to this kind of cartoon, okay, we see that when the diffusion layer is developed, we see that the bulk concentration is very different from the interfacial concentration. And since we are in electrochemistry, this is the interfacial concentration which basically defines the current we measure. And that's why in all the parameters now which we have here, this is the interfacial concentration which are involved and not anymore the bulk concentration. And to deal with this issue, what we can do is we can write the flux balances in the vicinity of the interface, okay, and then we can solve this either analytically or numerically. And what we can get is this dependence of the interfacial concentration on the bulk concentration. And as you can see, this dependence is highly nonlinear, which means that it's kind of impossible to disregard this dependence. But what we can do is that using this information, we can replot the calibration curve now as a function of the interfacial concentration, and only after that we can perform this graphical analysis as conventionally done in the enzymatic analysis, okay? And this can be done even more convenient with also classical Line-Weaver-Burk plot, which is a double reciprocal step. And so this was the first difficulty we encountered during this work. However, if we use the kinetics which we extract from this kind of calibration curve, even the corrected one, and we put it into the simulation of the release events, what we get is this green curve. And as you can easily see, it's far from being what we observe in the experiment. So the amplitude is very low, and the kinetics is also very sluggish. And to solve this issue, we thought about of several possibilities, but I will not mention all of them. The important point to mention here is that the outcome we obtained from the simulation already show us that we have a kind of problem with the kinetics of the enzyme. And so if we look how is diffusion is working in this case, so when the enzyme is sufficiently low in kinetics, okay, slow, when the glutamate reach the electrode surface and it saturates very quickly the enzyme which is on the electrode, it have to diffuse along the electrode or along the cylindrical part of the electrode to find the enzyme which finally can process this glutamate molecule. Okay, and if the enzyme is not really active, okay, the glutamate have to diffuse for sufficiently long times, okay, and this increases the probabilities that the glutamate will simply escape and will not be detected, okay, and this is evidently not what we want and basically not what we see in the experiment because the spikes are relatively sharp, okay, and it took us some time, okay, to realize the problem, but the point is that the pinpoint mechanism which I discussed above is generally written that this step with the creation of the intermediate product and intermediate enzyme is generally written as a reversible one. And it's crucial that this step should be reversible, and this is easy to understand because for the release events, it's not an important, it's not a big issue because the substrate is released in a very small quantities, okay? And so this means that these intermediate products will be present in the solution also in a very small quantities, and they can easily diffuse out, okay? But when we are in the situation of the calibration, the substrate is present in sufficiently large concentrations and large amounts, and basically this means that the intermediate products also will be in a large, present in a large concentration at some point, and at some point they will be enough to drive this reaction backward, and this immediately will affect the enzymatic efficiency, okay? Because part of the enzyme will work in the other, I would say backward, okay? And the efficiency in creation of the hydrogen peroxide will be compromised. And by adding this reversibility step, okay, this change the parameters which we can extract from the graphical analysis, and it adds also these two terms into the initial rate. Okay, but again, we can do with these equations, okay, we can still model the calibration curve and correct it with interfacial concentrations. And with the obtained kinetics now, what we can do is to fit really the calibration curve. In fact, the fit is even better than in the previous case. And also, when we model the release event under certain conditions, we obtain the curve which already recall by its parameters, by the amplitude and the kinetics, the one we observe in the experiment. And the two important points here is that if we compare with the amount of the glutamate which is released by the vesicle, okay, in the simulations, which can compute, this is represented by this blue curve, okay, again, with the kinetics, which we obtain with the modified mechanism, again, in a certain range of parameters, we can obtain the efficiency which is close to the 97%, okay, which is basically a kind of perfect efficiency for the enzymatic electrode. And just a final point is that, again, since we're in the simulation, we can construct a virtual situation where we can put the calibration curve for the release events. And so as you can see, due to the reversibility of this step, this calibration, how to say, for the classical calibration and would-be calibration curve for the release events, they are very, very different, okay? And during the release, enzyme appears as much more efficient than during the calibration when we have lots of the glutamate in the bulk solution. And to conclude, it's just with this modeling, we show that indeed we can have quantitative detection of the glutamate, okay, and this evidently supports the point of view that the glutamate release is sub-quantal, okay, but there are two other points, okay, that one should be extremely careful with the analysis of the calibration curve, and if the mechanism is not fully known, okay, you cannot basically perform and extract any quantitative information from the calibration curve. And any quantitative discussion like kinetic constant on sensor linearity range will be greatly affected, and so without doing this proper analysis, that basically is meaningless. And finally, I would like to thank my colleagues, in Paris, Professor Christian Amator and Dr. Irina Svir, the younger colleagues who were involved in this work, and evidently our collaborators in the Wuhan University, a group of Professor Weihua Huang. And thank you for your attention.